So membrane potentials. If we take any living cell, if we talk about animal cell, and we try to check electrical potentials on membrane using special cellular microelectrode, we impale cellular membrane, and second electrode we apply to the external surface of cellular membrane, and in voltage meter we can see presence of potential difference between intracellular and extracellular spaces. Potential difference looks like this. Inner surface of cellular membrane collect negative charge. Extracellular surface collect positive charge, relatively inner space of the cell. This potential difference we can call like resting membrane potential. How we can define it? Resting membrane potential is potential difference collected on the surface of cellular membrane in resting period, in value between -70 and -90 millivolt usually. So we can see some differences between cells of our body. Some cells has -90 in resting period, in other cells -70. It explains by the different properties of semi-permeability in resting period. Okay, so this property to collect and maintain potential difference in resting period is very important for functional properties of many animal cells. Especially it is important for excitable cells such as nervous and muscular cells, and for another cells too. Okay, so and our today's task to understand how this membrane potential is created, how maintains by the intracellular mechanisms, by membranous mechanisms, and first of all, we need to remember about properties of the cellular membrane. We know about semi-permeability of the cellular membrane. Some objects membrane can pass, another cannot. Generally, we can say about phospholipid bilayer that it is very effective electrical insulator. Cannot give ability for ions like anion and cations pass through this phospholipid bilayer directly. Any ion in water solution has water coverage that will disturb to penetrate between phospholipids of bilayer. For transporting of the ions through cellular membrane, we need specific structures like carriers that will make conformational changes of protein molecules for transportation against concentration gradient or down concentration gradient. Another type of the transportation is using ion channels with properties of the specificity and selectivity, with gating mechanism and without gating mechanism. So generally, for any excitable cell, we can describe this semi-permeability as following. Cell membrane of excitable cells such as neurons and muscular cells, are permeable in resting period for K+ ions. It means K+ ions without any problems can pass through membrane in direction of own concentration gradient. In resting period, cell collect K+ inside. That's why K+ using potassium ion channels, without gating mechanism, always diffuse outside the cell. It is outward current of K+. In resting period, membrane permeable mainly for these ion channels. That's why membrane potential depends upon this process mostly. But membrane is permeable for another ions, another cations such as sodium, calcium, but in very less amount. That's why influence of these currents to resting membrane potential is very, very low. Okay, so to the movement of the ion, to the ionic current through cellular membrane influence two main factors. Ionic current depends upon two factors. It is gradient of concentration, for specific ion, and second one is charge of cellular membrane. Because ion as a charged unit, when field action of the electrical field try to move in specific direction. Positive charged ions try to move in direction of the negative charge, and ions opposite direction of the movement. They try to be attracted by the positive charge. Okay, and these two forces, gradient of concentration, we can call this like chemical force, and charge, it is electrical force. When they act to the ion simultaneously, they influence each other. And finally, value of movement, intensity of the ionic current depends upon interaction between these two forces, chemical and electrical. Of course, these factors work in case of the high permeability of membrane for this particular ion. Okay? That will maintains only in case when ion channels are opened for this ion. For resting period, we have opened K+ ion channels. That's why these two forces influences to this ion in a resting period and achieves some steady state and in this steady state, we can see some value of the membrane potential. Okay, this value we will call like equilibrium potential for specific ion, in this case for K+. When sodium ion channels will be opened, membrane potential start changes and achieves new steady state between chemical and electrical forces, because movement of the ion through membrane will influence to the value of membrane potential. And when chemical and electrical forces will be equilibrated and movement of the ion through membrane will stop, will be achieved some another value of membrane potential. These changes of membrane potential and reasons influence to the dynamic of the process in process of excitation. We will talk about this later, and you will understand better these moments. Don't worry if you're not clearly understood right now. Okay, so what are the mechanisms we need to explain right now? First of all, we need to talk about terminology, some terminology that we will use in process of the description of these processes. First of all is diffusion potential. What is diffusion potential? Diffusion potential is potential difference that created on cellular membrane in moment of diffusion some particular ion through this membrane. For better understanding, we can see to this simple model that it is beaker consisting of two parts. These two parts are filled by the water solution, and in one of the part we dissolve some electrolyte, for example sodium chloride. In this solution present both sodium plus and chlorine minus. And between these parts we have semi-permeable membrane, like cellular membrane. Semi-permeable membrane that is selective for passing of positive ion channels of the cations. Semi-permeable can pass cations and not allow for passing of anions. Anions not allowed. So in this case, concentration gradient, relatively fixed law, wants to move these solute particles in direction of the pure water, in this direction. But membrane separate this movement. Chlorine ions cannot move. Okay? So we can eliminate this movement. Only sodium ions will pass through this membrane. Sodium will appear in this part of the solution. And when this ion moves through the semi-permeable membrane, this object brings positive charge from left side to right side. And on membrane will be appear potential difference. This solution will be charged positively, relatively left this side of the membrane, and solution will collect negative charge because anions will still positioned in this solution, and negative charge will be increases because deficit of positive will appear. Okay, so this separation of the of the ions will make diffusion potential. So this created charge on membrane we can call like diffusion potential. And actually cellular membrane is the diffusion potential is created by the ionic currents through cellular membrane that explains by the presence of the specific ion channels on the cellular membrane in resting period. This movement of the ions depends upon two factors, as we know: gradient of concentration and charge on the semi-permeable membrane. And we can see situation when diffusion of the ion will create conditions that will influence to the movement of the same ion. Okay, sodium create charge that influence to the diffusion of the sodium. Okay, so what is this influence of the charge to the sodium movement? As we can see, sodium brings what charge? Positive. When it moves through the membrane to the right side, it will make positive charge on the right solution. We can see interaction between charges: positive charge of sodium and positive charge of the solution. These types of the charges has same sign and will repel. It means electrical field of the membrane will disturb to the movement of the sodium to the right part of the solution. Okay, on the one hand, chemical driving force try to move sodium in right side. On another hand, sodium movement will be disturbed by the electrical field created by this movement. And when we leave this system to the some time period, we can see achievement of the new steady state when net movement of sodium to the both directions will be equilibrated. When chemical and electrical forces achieves equilibrium and value of sodium movement to the right and left sides will be equilibrated. And on membrane, we can find some value of the charge. This value of the charge has specific name. This potential difference we can call like equilibrium potential. Equilibrium potential for sodium ions. Equilibrium potential for sodium ions. Equilibrium potential depends upon concentration difference between left and right sides. In case of the cell, between intracellular and extracellular spaces, and can be achieved only when membrane is permeable for this ion. Okay, so as we know, equilibrium potential for sodium for usual excitable cells is around plus 40 millivolts. That's why when sodium ion channels are opened in cellular membrane potential start to move to this value, to the plus 40. If in resting period it was minus 70, minus 90, when sodium ion channels are opened, membrane potential want to have equilibrium potential for sodium and moves to direction plus 40. We can call this process like depolarization process. Okay? Equilibrium potential for the K plus ions for concentrations, for concentration difference for usual excitable cells of the human body, we have is around minus 90 millivolts. That's why in resting period, when K plus ion channels are opened in membrane in large amount, resting membrane potential is very close to this value. Okay, so equilibrium potential. How resting membrane potential are created? What are the mechanisms of the creation of RMP? Minus 70, minus 90 millivolts. We know two main mechanisms. First is ionic. Second one is metabolic mechanisms. Ionic mechanisms explains by the permeability of cellular membrane for ions in resting period. As we know, only K plus ions can pass through cellular membrane because of presence of non-gating ion channels for K plus that can selectively pass only K plus. That's why diffusion potential that's created in process of this diffusion will make negative charge on membrane. K plus ion is intracellular ion. Gradient of concentration as follows: inside more than outside. That's why K plus diffuse outside. And positive charge will go out of the cells. That's why inside cell has negative charge in resting period, outside receive positive. And the second one is metabolic. Metabolic mechanism depends upon active transport. Active transport using energy of ATP, and it is function of the enzymes. ATP is able to destroy ATP simultaneously transport two cations, sodium and K plus. We know this transport molecule like sodium, potassium, ATPase. Metabolic mechanism has very low direct influence to the membrane potential. We know about asymmetry of the ATPase functioning. Sodium, potassium, ATPase to the one cycle of the pumping against concentration gradient moves 3 sodium ions and as a response to one cycle pump inside 2 potassium ions. So one cycle will remove one positive charge for one cycle of pumping. That's why the more sodium-potassium pumps are active in this moment, the less positive charge inside the cell, and gradually it will make some potential difference on cellular membrane. But value of this is relatively low. There's only several millivolts can be achieved by this activity. Most important that are created by the sodium-potassium pump. It is gradient for concentration gradient for the K plus. For the K plus that will make condition for the ionic mechanism. As we remember, ionic mechanism based on the outward current of K plus. This current impossible without presence of gradient of concentration for K plus. So metabolic mechanism make two effects. First effect is direct action to the membrane potential making by the symmetry working pumping of the sodium and potassium. And second one that is most important for the maintaining of RMP is making of gradient of concentration for K plus. That will make condition for ionic mechanism. Ionic mechanism is very effective for creation of potential difference and main part of -70 created by the ionic. But this mechanism does not work without metabolic mechanism. Okay, it's such as secondary mechanism. Okay. For calculation of the values of equilibrium potential and membrane potential, we can use basic equations. First of all is Nernst. Nernst equation. Nernst equation allows us to calculate equilibrium potential for particular ion, as we know, most important for making of equilibrium for RMP is K plus ion. That's why we show RT, gas constant, absolute temperature, Z is the charge of the ion, F is Faraday's constant, natural logarithm, and concentration difference between outside space and intracellular concentrations. Okay, Nernst equation: gas constant, absolute temperature, charge of the ion, Faraday's constant, and natural logarithm of ratio of concentrations, concentration difference, a gradient of concentration between out and intracellular space. But we cannot directly transfer this equation for calculation of membrane potential because cell membrane permeable not only for K plus ions in resting period. Actually, we have a complex or combination of these permeabilities. So, and for calculation of membrane potential value, we have to use, it is more proper variant, RMP calculation. We have to use another equation, Goldman, Hodgkin, Hodgkin-Katz. Okay, or brief name GHK equation, and it looks as follows. Value of membrane potential is function of RT upon Faraday's constant, and we have to take natural logarithm of these values. Okay, so our cell membrane in resting period permeable for combination of ions, for sodium, for K+, and for chlorine. These three types of the ions make maximum value of the influences to making of membrane potential. And because of different permeability of membrane for these ions, we need to use this factor like permeability. PNA is permeability of membrane for sodium. And of course concentration, difference between extracellular and intracellular space. So, and same about K+ ions, K, permeability for K, and concentration difference between inside and outside. And the one negative charged ion, it is chlorine. Because of negative charge, we need to convert gradient ratio intracellular and extracellular. In, out. Okay, so this Goldman-Hodgkin-Katz equation that help us calculate value of resting membrane potential.